Hello, welcome back. It's Dr. Red Frizzle again. Right, today I'm going to be going into a bit more detail with the function and sort of structure of the kidney. In my last video, it was quite brief. It was just meant to be an introduction, so I don't get too worried if you're thinking, oh, I don't know all this stuff. There's loads more for me to remember because I'm going to go through that with you in this video. Right, okay, so first of all, we have our kidney here. I'm not sure how clearly you can see all these structures, but I've drawn them as best as I can, and I hope you can see them. If not, sorry, I apologise, it's the quality of my webcam. Right, so my second thing is I mentioned peritubular tubules last time, and I just wanted peritubular capillaries, sorry, and I just wanted to sort of finalise what that actually means. So basically, when you have your renal artery, so I'm just going to call that renal artery and renal vein, so I'll just label that RA and RV for those. So when you have your renal artery, renal vein, basically your renal artery, it sort of links up to these sort of small structures here. Now last time I sort of almost mentioned that the renal artery branched into the afferent arteriole, which then sort of filters out into this tiny structure called a nephron. But I'm not sure from my last video that you'll get a sense from the diagram how small these nephrons are. On average, a kidney has 1 million of these tiny little nephrons. I don't know if you can see that there. I've drawn it quite small because it is small. So you basically have 1 million nephrons on average per kidney. And in the nephron, I'll just do a quick recap. You have your proximal convoluted tubule. You have your loop of Henle. You have your distal convoluted tubule, and you have your collecting duct. Now, I'm not going to go into too much detail with the collecting duct in this video. I'm going to go into more detail with the structure of the proximal convoluted tubule lining. Right, so basically, so you have your glomerulus of your capillaries, which is your tight bundle of capillaries, which in case you can't remember, what happens is the renal artery, it branches off in something called the afferent arteriole. And the afferent arteriole is surrounded by hundreds of these, well, as I mentioned, 1 million nephron, which are all comprised of the same key components of these. So basically, what this diagram shows is it shows your glomerulus, which is a tight bundle of blood capillaries, and it shows basically your Bowman's capsule surrounding it, which helps absorb substances from the blood, in order to transport them to where they're required to go. So this is essentially the PCT lining. The reason why I'm going quite slowly through it in this video is because I really want you to sort of get a really good sense of where this is and to sort of help you understand, because I know there's nothing worse than watching a video that doesn't explain things properly. Right, okay, so this PCT lining has these little cells on it. So your PCTs being your wiggly bit. I've drawn that up here very small. I might just draw you a sort of quick diagram. So you have your Bowman's capsule here, your blood capillaries here, you have your PCT, which is your wiggly bit, and this essentially is just a bigger version at the very top of this bit. Okay, don't know if you can see that on this video, but basically it's just right at the top of this first sort of wiggly bit, which is called your proximal convoluted tubule. Right, so anyway, let's go into the structure of the proximal convoluted tubule cells, in order to establish how these substances are reabsorbed by the process of selective reabsorption. I don't know if you've come across that before, because I haven't mentioned it before. Selective reabsorption. Selective implying that it selects what it wants to reabsorb. It doesn't reabsorb everything from the blood, because there's certain molecules like red blood cells and proteins that want to stay in the blood. And how this is controlled is by this very advanced filtration system that's sort of in this renal or Bowman's capsule, as some people call it. Right, so first of all, you have your blood capillaries, and blood capillaries have an endothelium cells, which are these little, I don't know if you can even see them, but there's endothelium cells which line this Bowman's capsule. And basically, as you can see from this diagram, hopefully, they've got tiny holes between them, which enables the diffusion of dissolved substances in between these gaps. Your next layer is this bit here, so I'll just go over it in green pen just so you can see it. And this bit here is known as your basement membrane. Now the basement membrane only allows through molecules with a molecular mass less than, hang on, let me just make sure, I've remembered this correctly. It only lets through molecules with a molecular mass less than 69,000. So this is your sort of MR. So I'll write that for you. MR less than 69,000. Okay. So basically, once these sort of smaller molecules from the blood, like I've mentioned before, the proteins of red blood cells are not in this category. I can't stress that enough because these are big molecules, and let's do small ones, which I mentioned in my last video. I gave you a list of substances that selectively reabsorbed, and of course these are the molecules that actually can get through this filtration system. Right. So anyway, so the next step is these little sort of almost Pac-Man looking type things, and what they're called podocytes. I've actually read that there, you don't know if you see that, but podocytes. And what these podocytes, the reason why they're shaped with these sort of major processes, as they're known, is to establish these gaps between them. You might have seen this labelled as a foot process, but it's the same thing. It's these process-like projections, like coming out of them. So basically, once the glomerular filtrate, as it's called, because this whole thing here is the glomerulus. I probably should write that down for you. Glomerulus. Alright, so basically, once these substances have gone through here, they need to travel down this proximal convoluted tubule, and while they're doing this, selective reabsorption takes place. Right, so now is the important stuff, and I'm going into a lot of detail with this, so you need to be paying attention now. Right, okay, so basically you have your lining of your proximal convoluted tubule, so these cells actually have a sort of wiggly lining, which I've drawn down here for you. Right, so on this lining, it's folded to enable a large surface area as usual in biology. So lots of things need a large surface area, and it's especially important in the kidney because you want as much of these substances to be reabsorbed as possible. And these are called microvilli. And basically in these microvilli you have special proteins which I've sort of drawn as little boxes, but I'll just establish like little dots there. Right, so basically these are co-transporter proteins, and what they do is they transport the glucose and amino acids into this proximal convoluted tubule cell. Now you may be thinking, well, why do the glucose and the amino acids want to go into this particular cell? And the reason why is because the potential's been lowered by these sodium or potassium pumps on the opposite side of the cell. What these pumps do is they pump sodium and potassium into the bloodstream to lower the potential inside these proximal convoluted tubule cells in order for the glucose and amino acids to flow down the concentration gradient across the membrane of proximal convoluted tubule into the proximal convoluted tubule cells. Okay, so once that happens, then this lowers the concentration or the water potential, if you like. It increases the water potential, of the proximal convoluted tubule, because you've got all these substances that have suddenly moved out, and these are negative substances. So once they've moved out, the concentration gradient is increased, which enables water to flow by osmosis from in here into these cells again. So you've got this whole idea of glucose moving and amino acids that are diffusing straight through here by facilitated diffusion by these co-transporter proteins, and they end up in the bloodstream over here, in this region over here, which sort of resumes circulation as normal. And the reason why this takes place is because some of these substances that are squeezed out in this glomerulus, they're not all bad substances. There's a certain one called urea that's not selectively reabsorbed because it is actually a waste product, and your body does excrete that, so the kidneys don't selectively reabsorb it. And so this is where this whole idea of selective reabsorption takes place. Okay. Right, so once these substances have moved back into the blood, excuse me for that, once these substances have diffused back into the blood, in this proximal convoluted tubule, there's the next step of selective reabsorption, which happens in the loop of Henle. Loop of Henle being after your proximal convoluted tubule. It's the bit with the ascending limb and descending limb. Right, now in the descending limb, as you'd expect, the water potential is lowered. So we've got to sort of consider how water potential can be lowered. Water potential is lowered because ions are pumped out, essentially. So ions, you've got your K plus and your Na plus, which are sodium potassium ions, are pumped out. And the reason why they actually diffuse out. They don't need to be pumped out because it's down the concentration gradient, because this descending limb actually lowers the concentration. So once these ions are diffusing out, I should probably just mention quickly where this is taking place. the loop of Henle dips down into a region of your kidney known as the cortex, into the medulla, sorry. Because you have your medulla on the inside, and you have your cortex sort of almost around the outside, and then the membrane around the outside is actually called the capsule. Right, so basically these ions move into this medulla region in the inner part of the kidney, if you like. And what it does is it lowers the concentration, like to form a concentration gradient, which is vital for selective reabsorption to take place in the collecting duct. I'm not going to discuss too much about the collecting duct with you in this video, so I want it to be primarily about the loop of Henle and the proximal convoluted tubule. Okay, so in the loop of Henle, I've already discussed that these diffuse out here, higher up in this ascending limb. The permeability of these walls decreases. The permeability decreases, which means that they have to be pumped out by an active process known as active transport, which means that it requires energy from ATP in order to pump out these ions. So once they're pumped out, they lower the water potential in the medulla, which enables selective reabsorption to take place in the next step, which is after your DCT, distal convoluted tubule. You then have your collecting duct over here, which the water needs to flow back into the blood. But I'm not going to go too much into that. The other thing you need to know is when this water potential is being decreased, to decrease it even more, what happens is water flows by osmosis out of here as well. So this is water. Osmosis is down a concentration gradient. It's another passive process. It's quite similar to diffusion, but we sort of almost call diffusion of water osmosis on a regular basis. Right, so basically that's pretty much all you need to know about how the concentrations of these various ions are adjusted. I know it's quite technical with this PCT lining and the whole adaptations of it. I actually forgot to mention these mitochondria are in there. They're in there because these sodium and potassium pumps are essentially pumps. They are what they're called, which means they require a lot of energy to like carry out their pumping, essentially, to pump out these sodium and potassium ions out of this proximal lining cell. So that's why these have a lot of these mitochondria in here. I don't know if you can see this very clearly on this video, but I can assure you the proximal convoluted tubule lining cells do have a lot of mitochondria, even if you can't see it. So I hope this video has been helpful, and in the next video I'll be talking to you a little bit more about the permeability of the collecting duct wall. So I'm sure that'll be very exciting for you, so I'll see you then. Goodbye.